Good evening and welcome to the third Sir John Monash Public Lecture for 2015. The Sir John Monash Public Lecture Series provides Monash University, Malaysia an opportunity to engage the community in Malaysia and the region through the promotion of thought leadership in key areas that are strategic to the development of the region in the 21st century. This lecture series takes inspiration from Sir John Monash, who is considered to be one of Australia's most distinguished citizens, scholar, leader, and a man who sought to use his education and abilities for the benefit of the community. The public lecture series cover a wide variety of contemporary and multidisciplinary topics that will be presented by high-profile thinkers, international leaders, policymakers, corporate leaders, and leading scholars who are distinguished in their area of expertise and those who have made significant impact to the global community. The lecture series addresses key issues that are relevant to various stakeholders in the region on a wide range of topics, including the following: sustainability, socio-economic development, innovation and technology, health and well-being, societal transformation, Islamic economic thought and halal ecosystem, political transformation and international understanding. Today, we are very fortunate that we have Professor Donald Pfaff, who will present Sir John Monash Public Lecture on a topic titled Waking Up the Brain, Mechanism of Central Nervous System Arousal. Please allow me to present Professor Pfaff's biodata. Professor Pfaff graduated from Harvard College and received his PhD from Massachusetts Institute of Technology in 1965. He joined the Rockefeller University in 1966 as a postdoctoral fellow and was appointed as assistant professor in 1969 before becoming a full professor in 1978. His laboratory has published more than 900 research reports about how the brain regulates behavior. Professor Pfaff is a member of the National Academy of Sciences and a fellow of the American Academy of Arts and Sciences. Author of several books on the brain and behavior, he received the 2005 Award of Excellence in Professional and Scholarly Publishing by the Medical Science category from the Association of American Publishers for his book titled Brain Arousal and Information Theory. He also received the 2010 Aspen Foundation Prize in Neural Plasticity and the Daniel S. Lehman Lifetime Achievement Award in Behavioral Neuroendocrinology in 2011. Professor Pfaff is a faculty member in the David Rockefeller Graduate School and the Tri-Institutional MD PhD program. On a personal note, I know Don for 25 years. He works hard as he used to do. He works hard now as he used to do 25 years. And I won't be surprised when to fly back from here, he's going to go straight to the lab. He's very inspirational to me, and we had fun publishing a lot of work. I was with Don from around 1989 till about 1993, for 5 years in New York. So there's a lot I learned from Don. So, ladies and gentlemen, please join me welcoming Professor Pfaff, a great scientist, a great mentor, and a friend to the campus and Malaysia, and inviting him to present his lecture for this evening. Please.
Thanks, Professor Ishwar. It's an honor and a pleasure to be here, and thank you all for coming. So today's talk will come in 3 parts. The first part, describing generalized CNS arousal as a concept, the assay and the mechanisms for it. The second part will present a specific application of arousal systems in the brain, namely to explain how the chain of reproduction behaviors work. And finally, I'll have just a couple of slides linking the first two parts of the talk. Since General Sir John Monash was a civil engineer who applied his technical knowledge to problems of practical importance, I'll try to follow that philosophy in my talk as well. And thus, for each of the three parts of the talk, I'll try to state at the beginning the problem that we felt that we were facing. And then after I show you some data, I will say what the principle was that we think we've discovered. And finally, I'll attempt to relate that to the practical world outside the lab. Therefore, for the part one, the generalized CNS arousal, the problem is that we have the not aroused brain, the zero arousal brain. And that could be a patient in a comatose state. It could be a person who's deeply anesthetized, or it could be simply a person who is sleeping. And there are two questions that one could pose. The first is, can I come up with a concept that deals with all three? And the second is, can I discover the nerve cells which are the most powerful and essential for carrying out that concept, namely waking up the brain? And the answer to both of those questions is yes. So, about 10 years ago, as Professor Ishwar said, we said that there's a concept of generalized arousal which underlies all cognitive function and all emotional function. It's the most powerful and essential function in the brain. You can be aroused without being alert, but you can't be alert without being aroused. You can be alert without paying attention, but not the reverse. You can pay attention without the sustained attention that gives you learning and memory, but not the reverse. And you can learn things, but not be able to make decisions, which is a function of the prefrontal cortex, but not the reverse. At the bottom of all of this is this function that I theorized called generalized arousal. Likewise, with respect to emotional expression, whether you're expressing emotions on a lifetime basis, an hourly basis, or a minute-by-minute basis, the nature of the emotion depends upon how you're feeling and toward whom you're expressing that feeling. But the strength of the emotion depends upon arousal. And so, for example, if you were to think of emotional expression as a vector in physics, the angle of the vector depends upon exactly how you're feeling, but the length of the vector would be the strength of the feeling, and that depends upon generalized arousal. Now, for a concept which is supposed to be so important, you would think that when something goes wrong with arousal systems, many different medical problems or public health problems could exist. And that's what the most obvious, therefore, would be the stuporous state, the vegetative state, or the comatose state. But also the dementia of aging, the apathy of Alzheimer's disease, ADHD, autism, all have problems with arousal systems. Mood disorders, obviously depression, not enough arousal. Vigilance in the military or in shift work, the most dangerous shift work being in meat packing plants or fish packing plants. Dangerous occupations, toxicology, lead in the water would be one example. And finally, the mysterious fatigue states like chronic fatigue syndrome, fibromyalgia syndrome, Gulf War syndrome. All of these depend upon abnormalities of arousal pathways. Now, most of the time, your instinct will be to say that we don't have as much arousal in these various conditions as we would want. But in some cases, it's a reduction of arousal that is the thing that we're going after. And so the question is, can we anesthetize people properly? Yes, usually we can. And the reason that's in red is that I have anesthesiologists in my lab, and some of the data slides that I'll show you have to do with anesthesia. And sleep disorders. In the States, at any given time, 15 to 20% of the adult population has some kind of sleep disorder. So, arousal is obviously important. CNS arousal, crucial for all of these functions that I'm telling you about. But it was a bit of a sloppy concept, and I wanted to give it a strict operational definition, namely that a more aroused animal or human being is more alert to sensory stimuli in all modalities. The person or the animal is emitting more voluntary motor activity, and the person is more reactive emotionally. So we had to be able to measure this in a laboratory. We have a high throughput computer-controlled system. We have 32 of these in my lab. We have sensory stimuli for the sensory part of the operational definition, tactile stimuli, olfactory stimuli, vestibular stimuli. We're measuring every movement the animal makes 24/7 with 20 millisecond resolution. And for the emotional side of the operational definition of arousal, you could say, what kind of emotions can you count on in a mouse? And what you can really count on is fear. You can't necessarily count on existential despair or romantic love, but you can count on fear. And so we have a grid in the cage. We can shock the animal's feet and study either acute fear or conditional fear. So at that point in the development, several years ago, we had a new concept. We had an operational definition and an assay, but did we really know that generalized arousal exists? And we have three reasons for saying that it really does. The first would be quantitative behavioral evidence. Using a mathematical statistical technique called principal components analysis, we could estimate that about 1/3 of the variance is accounted for by generalized arousal. If you think of arousal as a partial differential equation, when you have a change in arousal on the left side of the equal sign, on the right side, there are going to be several terms. One of them is a generalized arousal term, and we're claiming that generalized arousal accounts for about 1/3, meaning that other forms of arousal, hunger, fear, sex, and so forth, and other factors that we probably can't even name, account for the other 2/3. You can't breed for something that doesn't exist, and we can breed for high arousal lines and low arousal lines of mice. Finally, you can't have mechanisms for something that doesn't exist, and we know a lot about mechanisms of arousal in the central nervous system. So I'm going to give you a very jet speed, high-flying tour of CNS arousal mechanisms. Some of you who are medical doctors will be remembering some of this from medical school. First we'll go through the neuroanatomy, then we'll mention some neurophysiology, and finally we'll talk about functional genomics. People who have gone to medical school know that we have ascending arousal systems: norepinephrine, dopamine, serotonin, histamine, and there are acetylcholine neurons in the basal forebrain as well as in the midbrain, which invest the entire cerebral cortex. All of these arousal systems, ascending arousal system, have their cell bodies farther posterior in the brain. The axons go forward and wake up the anterior part of the brain. Most people don't think of axons going the opposite direction, but they do. From the hypothalamus, the lateral hypothalamic area, come the orexin. Well, one lab calls it orexin, the other lab calls it hypocretin. The fact is that if you have a mutation in this gene, then you're going to have narcolepsy. Narcolepsy means that you're at a party, you're laughing, especially laughing and showing some emotional expression, and all of a sudden you have total postural collapse and you're lying unconscious on the ground. That's a result of mutations in the orexin gene. The paraventricular nucleus of the hypothalamus, the oxytocin and arginine vasopressin. The histamine neurons have descending axons as well as ascending, and there are neurons discovered by Arthur Lowy in St. Louis where axons go all the way from the basal forebrain, the preoptic area, all the way down to the spinal cord, because all of you know that a part of arousal is the autonomic side of your body, that is your heart rate going up, respiration rate changing, sweating in the skin, and blood pressure going up. So this descending system is part of arousal as well. This is all well and good. But what fascinated me is that about 40 years ago, a Spanish neuroanatomist discovered cells who have axons going in both directions and have the capacity to participate both in ascending arousal systems and in descending arousal systems. And these neurons are in the back of the hindbrain. They're just above the spinal cord, beneath the cerebellum. And I'm picturing several of them here. Here you have the black cell body. You have the axon coming out of the cell body. You see these red arrows. And then at that point, the axon bifurcates. And one of the axons goes toward the forebrain, and the other axon goes toward the spinal cord. We think that these large neurons, which I'm going to argue are the most powerful and essential for arousal, have the capacity both to participate in ascending arousal systems and also in descending arousal systems. Here's another one over here. I love these neurons. Here's the big cell body. They're among the largest cells in the central nervous system. The axon comes out, crosses the midline, bifurcates at this point, with one branch going toward the cortex and the other branch going toward the spinal cord. These cells are so large that they're called giant cells, and the group of them is called nucleus gigantocellularis, NGC. And a nervous system signal talking to nucleus gigantocellularis, NGC, is a little bit like an American politician talking to Bill Clinton. Once you've talked with Bill Clinton, you've talked with everybody. And these guys are talking with everybody. They have their axons going up to the thalamus. They have axons going forward to the pons and to the midbrain as well as to the basal forebrain. And as I said before, they have axons going to the spinal cord, all levels of the spinal cord with terminations bilateral. So these are very, very well connected neurons. They have the power to underlie generalized arousal. What about the physiology of these cells? Eugene Martin and I, several years ago, said, what do we expect of these cells if these cells really are important for fundamental generalized CNS arousal? And the 5 criteria were that they should be responsive to all sensory modalities, they should habituate, namely, if the same stimulus is repeated time after time, the magnitude of the response should go down. They should drive cortical electroencephalography, they should drive EMG, that's muscle electrical activity, and they should be associated with the activation of behavior. That is, that if the animal is standing there still, and just before the animal moves, these neurons fire. And the answer for all of these criteria is yes, and I'm going to illustrate just 2 of them in order to stay within time. Here's responses of an individual nerve cell. What you've got here is a cross-section through the hindbrain just above the levels of the spinal cord. And this particular neuron is in the reticular formation, and it's responding. Right here is the onset of the tactile stimulus, several repetitions of it. Here's the onset of the olfactory stimulus. I'm going to come back to visual. Here's the response to the vestibular. Here's the response to the auditory. And you'll notice also that the response tends to fade. It habituates. And the visual is more confusing because the cell obviously can respond. Like here's no spontaneous activity, and then the cell responds at the beginning of the stimulus. Here's 0 point right there. Here again, and here again, and here again. But when Eugene was looming over the animal, some biological behaviorists said that he was acting as a visual stimulus himself as he came close to the animal. So we think that this is actually the response to the image of Eugene, the experimenter, rather than the light that he was flashing in the animal's eyes. What about perturbing these? In other words, we can record from these cells and we can connect them to arousal. But what about if we perturb them? Can we get changes in arousal in the cortex or in behavior? And the answer is yes. Here is a sleepy EEG. Remember, EEG stands for electroencephalography, and that would be the electrical recordings from the surface of the skull. And in this animal, which is deeply anesthetized, if we miss the NGC, off-target stimulation, we have no change. But if we actually stimulate NGC properly and electrically, we change from the sleep EEG, which is low frequency, high amplitude, to an awake EEG, which is high frequency, low amplitude. I mentioned anesthesiology. Is it possible that we can prove that when an anesthetic is taking action and quieting down the EEG, and for that matter quieting down behavior, that it's acting through NGC? And so what Dr. Calderon in my lab did in this experiment is that she had a very wake EEG, and then she injected an anesthetic, isoflurane, directly into the nucleus gigantocellularis, and it stayed there. It was fluorescent, so you see it right here. Here's the cerebellum, here's the brainstem right here, and the fluorescent anesthetic is right in NGC, and at that point she gets a sleep EEG, and then 10 minutes after post-injection it wears off, and we have an awake EEG again. I won't bother to describe that. Now, these anesthetics that I'm telling you about work through GABA receptors. Gamma-aminobutyric acid, GABA, is an inhibitory neurotransmitter. And the anesthetic turns on the inhibition such that the NGC can't operate and the animal goes to sleep. So what she's got in this case is a deeply anesthetized animal, and she puts in a GABA blocker. In other words, she takes the inhibitory neurotransmitter and she blocks the receptors. When she blocks the receptors, only in NGC, she can get the EEG to wake up, and then 20 minutes after, the GABA blocker wears off and the animal's deeply anesthetized once more, proving that that the NGC is necessary and sufficient for anesthetizing the cortex. What about the genomics of this? Does the nucleus gigantocellularis express the genes that you would expect if it really is important for generalized arousal? And the answer is yes. This is from a polymerase chain reaction in which the results, as a result of the number of cycles of the reaction, the amount of product goes up. And the threshold is here in red lines. So what we're looking for is the gene products to go above threshold. And here you have remember I told you about orexin. Here you have the orexin receptors going above threshold. And remember, opioids will decrease arousal. And here's the delta opioid receptor and the kappa opioid receptor, as well as the mu opioid receptor, all going above threshold. So the receptors that manipulate arousal, either increasing, like orexin, or decreasing, like an opioid, are expressed in these neurons. The neurons are expressing genes that they should be expressing if they are doing the job for arousal. So wrapping up this first part of the talk, what we really know is that these reticular neurons, NGC neurons that I'm telling you about, are crucial for managing the arousal of the entire central nervous system, of the cortical arousal and behavior. What we need to know now is a molecular characterization of these neurons. We're producing these neurons from embryonic stem cells, and so we'll know a lot about them actually in about 1 month. Further, all of our brains have a blood-brain barrier that prevents nasty stuff, which is circulating in the bloodstream, from getting into the brain. But it looks to us in a very small number of cases as though some of our neurons, some of our NGC neurons, are outside the blood-brain barrier, that the blood-brain barrier is leaky and that things can get from the blood into these neurons. And if that's true, that's very important from the point of view of medical manipulation. So what we're trying to do now is to verify those very small numbers of cases. We don't know yet, actually, if it's a small story that's going to turn into a big story with further effort, or whether we're chasing a permanently small story and that we're wasting money and time. But in about a year, I'll know the answer to that question. So what have we solved in the first part of this talk? What we solved, the principle that we have, is that there's a restricted number of neurons deep in the hindbrain, just above the spinal cord, which are essential and powerful for manipulating arousal of the entire brain. And the reason that's important is it tells us the neurons that have to be manipulated in the future for advanced medical manipulation of patients in a comatose state, patients in a vegetative state, patients with sleep disorders, and patients with traumatic brain injury. So I look forward to, I'm not a medical doctor, as you know, but I look forward to medical application of the basic science that we have in the first part of this talk. The second part of the talk dwells now on a specific form of arousal, namely sexual arousal. And the problem is that many neuroscientists have concentrated on learning and memory. But as David Anderson, who's a professor at Caltech, says, a central problem in neuroscience is to understand how instinctive behaviors are encoded in the brain. And when you stop to think about it, although all of us want to be smart, I know that tests started at Monash University just today, and we want to get as high marks as possible. But a lot of the things that make life worthwhile, love and sex and fear and hunger and thirst, they're all instinctive behaviors which neurobiologists have sometimes let go in the interest of studying learning and memory. So what we've done is we've taken a chain of behaviors, courtship followed by copulation followed by maternal behaviors, and we found out the mechanisms which are essential for reproduction, and we found them out mainly on the female side. And we've determined that an individual gene, estrogen receptor alpha, ER alpha, expressed in a specific group of hypothalamic neurons is essential for these sex behaviors, and the same gene expressed in the preoptic neurons, I'll show you the data, essential for maternal behaviors. So what's the proof? Here what you have is a very simple animal behavior. We figured it had to be simple because the mammalian brain is really complicated, and if you're studying a complicated behavior and a complicated brain, chances are you're going to do a lot of weeping. And all the four-footed female animal, whether it's a rat, a mouse, a hamster, a cat, a dog, has to do is to allow the male to fertilize. And by explaining how this works, what we showed is how steroids coordinate brain function with the rest of the body in order to ensure reproduction in an appropriate environment. And putting it from the point of view of physical measurements, we understand these mechanisms from angstroms, which is 10 meters times 10 to the minus 10th, up to seasonality, which is measured in light years. And the proof comes in 3 steps. First is discover hormone receptors in the brain. You're looking at a rat brain from the left side, and each dot is a group of neurons that express this estrogen receptor alpha, and what you have is a limbic hypothalamic system, which is universal among vertebrates, from the fish to the philosopher, reptiles, amphibians, birds, higher primates, human brain. This pattern of basic forebrain, that is limbic forebrain, and hypothalamic expression of these sex hormone receptors holds true. But for today's talk, the most important are all squeezed down here into the part of the hypothalamus called the ventromedial nucleus of the hypothalamus. And the reason that it's important for today's talk is that discovering hormone receptors in the brain was one entry into being able to unravel the neural circuit for an instinctive mammalian behavior. It's the first one that was ever discovered. So our 3 points of entry to discover this neural circuit were first the estrogen receptor binding neurons that I told you about just a moment ago up in the hypothalamus. The second would be the cutaneous stimuli which trigger the behavior, and using electrophysiology and neuroanatomy, we could follow the signal into the spinal cord, up the spinal cord, into the brain. And finally, we could take the muscles that actually execute the response, which is called the lordosis response. These are deep back muscles, which are run by these motor neurons here, and these motor neurons receive descending signals from the brain. And by triangulating from the sensory side, from the motor side, and from the endocrine side, we could discover a spinal, midbrain spinal circuit, which is controlled by an estrogen-dependent signal coming out of the hypothalamus. And it took, I knew that it was going to be a lot of work, but I didn't know it was going to take 20 years to put it together. But it was worth it, because at that point we got lucky. It turned out that the estrogen receptors, and for that matter other hormone receptors that we had discovered in the brain, are ligand-activated transcription factors. A transcription factor is a protein that turns on genes. And ligand-activated transcription factor means that it will only turn on genes if the hormone is on board. So the hormone comes, binds to the hormone receptor goes to the surface of the DNA and turns on the gene. And I'm going to show you how that happens in a couple of minutes. But first I want to show you the behavioral data. Oh, no. I'll show you the genetic data immediately. What it turns out is that if estrogen binds to either the one I told you about, estrogen receptor alpha, or ER beta, then you have a series of genes which are turned on. Their messenger RNA levels are increased, except for this one down here, and the gene product drives female reproductive behavior. This one down here works by disinhibition. The hormone turns it off, and that's important because the gene product turns off the behavior. Now the question is how this happens. And now that I'm back in order, I'm going to tell you in a couple of slides how it happens. But first I want to show you the raw behavioral data. If you use a viral trick to turn off gene expression for estrogen receptor alpha only in the hypothalamus neurons that are necessary for behavior, the behavior goes from zero. Here the animal is set up with estrogen so that the animal can do lordosis behavior, but you knock out that gene and you have zero behavior. We replicated that in the next experiment, and what the female is doing instead is rejecting the male. So when you knock out ER alpha, the estrogen receptor alpha, then the rejection behavior goes up. Likewise for maternal behavior. What the mother mouse or the mother rat, or for that matter lots of other animals, is supposed to do is if the little baby pups, in our case baby mice, are spread around the cage, the mother runs around and very gently picks them up and brings them back to the nest. That's important because they have to be kept warm, they have to be fed, and they have to be kept away from predators. Furthermore, she licks those babies so that they stay moist, and she nurses them. And if you knock out estrogen receptor alpha in a specific group of neurons called the preoptic area neurons, then the latency to do this pup retrieval goes almost the total length of the test, which is 300 seconds, 5 minutes. The licking goes down and the nursing goes down. And the effect on maternal behavior, and obviously we know that maternal behavior is crucial for people as well as for mice, but the effect was so strong that it was covered on American television. And I was accused on network television of discovering the mommy gene. If you knock out this this gene, then mommy doesn't behave like mommy anymore. And the commenter, who was not a scientist, said this is a very bad thing because now mothers that are just naturally lousy mothers are going to blame it on their mommy gene because they found out from our paper about the strength of this effect. So the behavioral effects are crucial. And I'm going to make a point at the end of this section just how crucial this system is to study. But let's get back to the technical side of it. We have the hormone coming into the brain, liganding, that is binding, to hormone receptors, and then genes being turned on. And the question is exactly how does that happen? And here's how. You have your DNA, which you all know about, and it's an inactive DNA. Most of the DNA is inactive most of the time. And the way it's kept inactive is that it's wound around these proteins in 146 nucleotide bases per nucleosome. Each little barrel here is called a nucleosome. So here you'd have about 146 nucleotide bases inactivated, 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 inactivated. But through the actions of hormones in a hormone-responsive system, somehow that DNA gets free, such that the transcription factors I've been telling you about and their companion proteins called coactivators can get in there and turn on the gene. Now, the simplest way of understanding how this happens is to look at the nucleosome from the side, and that's what we're doing on the next slide. And so now we're looking at from the side, and what you have in there is proteins which are called histones. And most of the chemistry of this system was discovered actually in yeast, but it's conserved into animals and even into mammals and human beings. And these histone proteins have their N-termini sticking out. And the N-termini are susceptible of chemical modification. And the chemical modifications are called methylation or acetylation or immunation or phosphorylation or combinations of the above. And what we're most interested in is modifications of these histone tails which allow transcription to be facilitated. Let's call them pro-transcriptional events, because we know that the hormone, when it goes into the hypothalamus, has to turn on genes, has to synthesize messenger RNA, has to synthesize new proteins in order for the behavior to occur. So we're trying to discover modifications of histone termini which will allow the nucleosome to fall apart such that the transcription factor can get in there and turn on genes. And we have done that. So especially how it links out is that you have the estrogen receptor with the estrogen on board, and once it's on board, it comes to the nucleus and has a DNA binding domain, DVD, DNA binding domain, and it binds to a specific sequence on DNA, thus to turn on estrogen-sensitive genes. Here we have estrogen. If we had a stress hormone like glucocorticoids, we would talk in human endocrinology about cortisol. We all know what cortisol is. So cortisol can bind to the glucocorticoid receptor. It also has a DNA binding domain. And you'll notice that this sequence looks fairly similar to that sequence, but it's not the same. This goes AGGTCA, this goes AGAACA, and so forth. So the different hormones with their different hormone receptors are kept separate from each other by virtue of their DNA response sequence. Now, we do have the protranscriptional event because estrogen treatment acetylates histone number 4 in hypothalamic neurons. Here we have the immunocytochemistry for the pan-acetylation. Here we have ER alpha. When you put these two together, you have yellow cells, which are cells that express ER alpha and are acetylated, a protranscriptional event. The trouble is that with that immunocytochemistry, you can see in exactly which cells, the behavior-relevant cells, the acetylation occurred, but you can't tell which genes are activated. The way you do that is that you have an antibody which is specific to the altered tail of the histone, chemically modified histone tail. You do a technique called immunoprecipitation to get all of these out of the solution, and then with that PCR technique I told you about before, you recognize the specific genes which are about to be affected. And I can show you some data. It turns out that the hormones do cause these histone modifications which will turn on genes, but exactly what happens depends upon the histone mark you're studying and the promoter segment that you've picked out. So here we have the promoter segment of an estrogen-responsive gene, and we're interrogating these three parts of the DNA. This is a string of DNA sketched here, and we're interrogating these parts. And with the acetylation, yes, indeed, you do have an increase, sometimes statistically significant. But with another pro-transcriptional event, histone 3, lysine 4 trimethylation, you have a different pattern of results, such that on a different part of the promoter, 5 hours after estrogen, you have the significant increase. And likewise with the oxytocin receptor, which is also hormone sensitive, we interrogate different parts of the DNA. We have the increase after estrogen treatment at 3 hours, here and here, but with this different histone mark, also pro-transcriptional, you get a different pattern of results following estrogen treatment. So it's complicated. I wasn't looking for it to get any more complicated, but it did, because a fiery, strong-willed Brazilian student came to the lab and she said, you want life to be simpler, but I'm about to make it more complex. Because it turns out that thyroid hormones interact with our system. Everything I've been telling you up until now is estrogen, 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 female hormone, female hormone, female hormone. But it turns out that thyroid hormone receptors, here in the upper left of the slide you have T3, which is thyroxine, they bind to the same sequence as estrogen receptor, only part of that sequence. And we hypothesized, under the influence of this Brazilian student, whose name is Larissa Costa Faustino, we hypothesized that giving thyroid hormone would interfere with the estrogen effect of the sort that I've been showing you for the last 2 slides. And it's a very complicated slide, and so I'm going to make my main point, tell you both my victory and my defeat, or let's say my victory and Larissa's defeat, in this experiment. And here's what it is. That as I've been showing you in the past couple slides, when you give estrogen, you can get an increase in the acetylation of histone 3, which is a pro-transcriptional event. It favors transcription. And we hypothesized that when we give thyroid hormone as well, that effect would not occur. So when we made the animals hyperthyroid, sure enough, we didn't get that increase anymore. We had that as a significant increase, but here it doesn't happen. That is exactly according to theory. The trouble is that when we made the animal hypothyroid as a control, it also happened. And so this is directly in favor of the theory, and this is directly against the theory. So we have a tremendous amount of data on this slide, but about 50% of it favor my idea, or favor Larissa's idea, and about 50% don't. So life was getting so complicated that I wanted to do something different. Instead of observing changes in the brain, I wanted to go in there and cause changes in the brain. And when I would cause the change, would I get a subsequent change in behavior? So what Zachary Weil did in the lab is to set up the animals so that if we put just artificial cerebral spinal fluid, ACSF, into the hypothalamus, that we wouldn't have any female reproductive behavior at all. But if we put in ACSF plus a deacetylase inhibitor, a deacetylase inhibitor leads to more acetylation, then we should get lordosis behavior, and sure enough we did. In 40% of the animals, we got lordosis behavior, and the rejections went down. So you can get the drift from the past 5 minutes or so of this talk, that when we've gone in and tried to observe the neurochemistry, it's been terribly complicated, and we don't always get what we think, what we predict we're going to get. But if we go in and manipulate the neurochemistry and look for behavioral changes, that seems to be a more clean way to do it. So what we've got is a specific gene, estrogen receptor alpha, and specific neurons that act out through a determined circuit, the first circuit for any mammalian behavior discovered, to regulate a specific behavior, a female reproductive behavior, that's essential for fertilization. Encoding the chain of behaviors by ER alpha shows us how mechanisms for instinctive behaviors can work and how they're harmonized with preparations for reproduction elsewhere in the body. Because after all, in both males and females, there have to be changes elsewhere in the body for reproduction to occur. And what we're doing right now is this chromatin immunoprecipitation at Rockefeller using genes of neuroendocrine significance. And we have a balance between genomic mechanisms and non-genomic mechanisms of hormone action. So the principle that I can say to you now is that yes, if you pick a vertebrate behavior, a mammalian behavior that's simple enough, you can discover exactly how it works. Now, why is this particular, coming back to the application to a problem of significance, important? And the reason that it's important is that we probably already have exceeded the carrying capacity of the Earth. Right now we're about 7 billion, and many people say we're headed for 9 billion. The Population Council in the States says that we're not likely to get population under control during this century. And we have these droughts, we have extreme weather, we have people starving all over the place. And so understanding reproductive biology and limiting reproduction in a reasoned and ethical fashion, I think is going to be essential for the future of the species on the planet. And so I look forward to reproductive biology broadly conceived being applied in an enlightened way, probably in every country on the planet. So I've given you a lot of data and thinking about a very general brain-wide function, namely generalized CNS arousal. And I've also given you data about a very specific instinctive behavior, namely female reproductive behavior in a laboratory animal. It turns out that these two things are related. In the laboratory of my former protege, Jessica Maughan, she showed that pharmacally heightened arousal elevates female rat courtship behavior. Secondly, genetically lowering arousal reduces mating behavior. We did that in our lab. Thirdly, we know exactly how these things work. In the hypothalamic neurons, which are essential for the behavior, arousal transmitters turn on these neurons, and new opioids turn them off. In the preoptic neurons, essential for lordosis behavior and maternal behavior, a somnogen, prostaglandin D synthase, reduced by estrogen, and in that case, we're reducing something that inhibits the behavior. And finally, when you look at these behaviors just in videotape of animal behavior, it's evident that they require high generalized arousal. Fast starts, rapid locomotion, fast stops. So on the female side, it's evident that generalized arousal is essential, is connected with sexual arousal. And intuitively, you would think that. Likewise for the male. In this slide, all of the important data are on the left side of the slide, and we sorted the data according to parental arousal, or the arousal of the animals we're actually testing. This is in our breeding project. And here, sorted according to parental arousal, or arousal of the animals who we're actually testing. And it turns out that the animals which are bred for high arousal, when you just put the female in their cage, they go nuts. They're like teenage boys on their first date. They're not doing what they're supposed to be doing. They're not doing what their parents expect them to do. They are really highly motivated, but they lack skill. And so they jump all over the female, but they don't know what to do. And when they finally achieve insertion, they ejaculate right away. Whereas the low arousal animals do exactly the opposite. They are not excited to begin with, and finally it takes them forever actually to reproduce. So it turns out that you can breed for arousal. You can make a prediction going from generalized arousal to sexual arousal, and link the two. And the reason I emphasize the Maughan lab so much is that she now has her own lab at University of Maryland, and so all the data on the female side were from her lab, not from my lab. So the import here is that most of the things that people really care about, and as I said at the beginning of the second section, have to do with these instinctive primitive behaviors. Sex, hunger, fear, all of the emotions that make life either worthwhile or, in the case of fear, go opposite what we're hoping for, are these instinctive things. And you think of them as separate entities. They have connected with separate emotions, but actually they're linked. And so sex and fear, or hunger and thirst, they're woven together in systems that make biologic importance. And in this particular part of the talk, I simply wove together generalized arousal and sexual arousal, simply because those are things that we know a lot about. We could have done it with other systems as well. So in summary of the entire talk, we have a primitive brain function, generalized arousal. We hypothesized it, we defined it, and we assay it. We know a lot of its neuroanatomical and neurophysiological mechanisms, and we're deep into studying its genomic mechanisms. We're now using embryonic stem cells to produce the cells that are essential for generalized arousal. We highlight its quantitative properties. Remember, we're measuring the animal several times per second. And what we're really interested in is application to medicine and public health. And I know that some of you in the audience are as well. Heightened states of generalized arousal. The easiest ones to talk about in experimental animals, are sex and fear. We know those are things that they do. And what we did is to determine how simple sex behavior is regulated and how we study its relationship to generalized arousal. Obviously, generalized arousal contributes both to male and female sex behaviors. And what we're really interested in, I come from a doctor's family and I'm allergic to mice, so we have to study mice because they're the experimental animal of choice, especially for genetics. But what we're really interested in is the applications of these facts about generalized arousal to medical problems. At the beginning of the talk, I told you a laundry list of medical and public health conditions, and especially those with large sex differences. The fatigue states, for example, females are much more preponderant to get those chronic fatigue syndrome or fibromyalgia syndrome than males. But in other cases, ADHD or acting out, all of the crimes of aggression tend to be more males than females. And so we can put our knowledge about sex differences in the brain to a special use with, for example, depression, female dependence, ASD, and ADHD, more typical of males. So as you can guess, I'm representing a large group of people. For these particular data that I showed you today, my longtime associate, Dr. Li-Meng Cao, is a biophysicist, and he's essential for a lot of the data. A molecular biologist named Katuna Gagnidze produced a lot of the data. Also a real dynamo in the lab, Rebecca Leshan. And the woman who caused so much trouble, Larissa Costa-Falsino, she made our life more complicated when we were looking for it to get simpler. She's happy. She's a happy camper. And she's back in Brazil. She's earned her PhD, and she's making her career in Brazil. So this is the summary of the talk. Although I welcome questions on all points of the talk, I'll also remind you, as Professor Ishwar said, that I've been doing this for over 50 years, and I know a lot about the regulation of behavior by the brain. And so any scope of question that within brain and behavior relationships, I'm happy to try and address. Thank you very much.
Yes, Don. Thank you very much for that very arousing talk. Now we are open for questions. Any questions? Let me start first. Don, you talked about steroid hormones, particularly estrogens and all, and some neurotransmitters. You didn't mention about neuropeptide S, which is also involved in arousal. How do you think it integrates into this circuitry, neuropeptide S?
We know it's expressed in hypothalamic neurons, and we know that it's expressed in nucleus gigantocellularis, but we don't know exactly what it's doing.
Right. Any more questions?
Hi, my name is Siketo. I am actually interested in information theory. Sorry. Okay, you mentioned about the instinctive behavior of animals and things like on humans as well. Is it only based on hormones and their sensors, or are there other things that would affect them?
Things that affect generalized arousal. It's certainly not dependent exclusively on hormones, no. All kinds of exciting stimuli from the environment will raise arousal, raise the animal from sleep, and so forth and so forth. The reason about information theory in the title of that book, Brain Arousal and Information Theory, from about 10 years ago, is that we think that it's surprising stimuli which wake up the brain. And so information, as you know, information is a measure essentially of improbability. If we don't expect the stimulus to occur, and it does occur, then it has a high information content, and it's much more likely to wake up the brain and stimulate behavior. Whereas if the stimulus is repeated time after time after time, like a metronome that you could use to time your piano playing, then it becomes extremely boring and does not arouse the brain at all. So large numbers of stimulus environments, and also I might say drugs, will affect the arousal systems.
Thank you.
I'm Anthony from Sunray University. We hypothesize that the nucleus Generalized as an important nucleus for in comatose patient upon electrical stimulation.
Is that a question?
Yeah, the question is whether the nucleus gigantocellular is it important for
Nucleus.
NGC?
Yeah.
Is it important for comatose patient?
Well, I have to be careful because I have to split my answer into two parts, as we were talking about before, to a neurosurgeon who's sitting right here. On the one hand, I'll speak as a neurophysiologist and a theoretician, and on the other hand, I'll speak as a person who does not do clinical work himself. I'm absolutely certain, as a theoretician and a basic laboratory scientist, that manipulation of cells in nucleus gigantocellularis would have a huge effect on vegetative state patients. Not comatose patients. Coma is by definition a temporary state. A comatose patient, let's say it's the patient who crashed his head on the rock and the blood has gotten loose and is pressing on the brain and will kill the patient soon. That comatose patient either will die or the comatose patient will go into a vegetative state. So what we're really talking about is the vegetative state. I am certain that electrical stimulation or other means of waking up these NGC neurons would be therapeutically useful in such patients. That's the first part of the answer.
Has anyone done that before?
Wait. The second part of the answer is that those nerve cells are so crucial for the processes of life that if the doctor makes a mistake, either with his electrical stimulation or with his manipulation of the electrode, because the electrode does damage, he's likely to kill the patient. And so the neurosurgeons that I know, in New York City, for example, if I were to say to them, yo ho ho, we've done a lot of work with mice, and why don't you apply this to your patients tomorrow? They would look at me and they'd say, you have to be crazy. So I'm confident that in the long run, in the progress of science and medical science, that we'll think of ways of manipulating NGC neurons in ways that are regulatable and safe. That is, that the benefit of doing so for the vegetative state patient is much greater than the cost of making a mistake. We know, and we talked about this in the Malaysian situation earlier before the talk, that the human cost, the emotional cost, the economic cost of maintaining these patients indefinitely for ethical reasons is severely great. It's just terrible on the family and terrible for society. And so you not only have the technical questions of how do you avoid that excruciating system, but circumstance, but also you have the question, the ethical question of how do you make decisions about such things. And in my experience, which is not clinical experience, but it's just reading what other people think about, is that individuals never make such decisions. That it's always going to involve family members, it's going to involve medical personnel, it's going to involve religious personnel, and it's going to involve a certain amount of philosophical complexity. And those people looking at each individual case will have to make these decisions. And that's far beyond the capacity of an individual neurobiologist like myself to say anything.
Thank
you, Professor Pfaff. I think it's an excellent talk. I saw in your slides a number of times that you've actually listed down ADHD and also autism.
Yes.
Over the last 2 decades, we're seeing increase in ADHD and also autism. Mm-hmm. From your own research over the last 50 years, I'm sure that you've seen the patterns of the various diseases. What could be, from a theoretical perspective, could it be the hindrance of the flow, or the highways that you're showing, both the north and south? Mm-hmm. Are there, are there the disruptions are due to external, or is it something that's happening that is within our own genetic mutation that is causing this sudden increase across the globe?
So it's a really important question about ADHD and autism, and really a complicated set of answers. And so I'm thinking about approximately 4 dimensions at once in trying to come up with the answer conundrum. Let's talk about ADHD first. ADHD clearly can be genetically influenced. There's a number of repeats in the dopamine 4 receptor, and the number of repeats in the promoter of that receptor, of the gene for that receptor, determines whether or not the person is likely to get ADHD. On the other hand, it's environmentally determined as well. And there's a sex difference. More boys are diagnosed with ADHD than girls. And there's an environmental thing here. That is that once the teachers and society in general learn the 4 initials, ADHD, and you have a kid in class who's a pain in the neck, then you're more likely to assign that child to a special class because that child has ADHD and get him out of your hair rather than put him up with him. So there are strategic things in the teaching profession that may affect the diagnosis. And when we talk about rates of diagnosis going up, it could be that we're getting it more, but it also could be that the teachers have figured out how to get those kids out of class. Finally, is it really the child who is the problem, or is it the requirement of keeping active young boys sitting on their backsides for hours and hours that's the problem? After all, if we were in a farming family, we would be out there working, and then the teaching and learning would assume a certain number of hours, but not the way we have it now. And so some experts in ADHD are saying, yes, there may be sickness in the child, but there's also sickness in the school system. And so this guy, who's a professor at University of California at Irvine, has special schools for ADHD where they're up and running around a certain amount of the time and then coming back and sitting down. And by the manipulation of the teaching environment, they're able to reduce the impact of the genetic mutation. Okay, what about autism? That's again a complicated question, as you hinted at in your question, Mahendran. The rates of diagnosis may simply be that people are noticing things that we used to be able to say, oh, that little boy is a bit odd. He's a little bit socially awkward. But now we can diagnose him with something, and it's a medicalization of a problem with normal human variation. After all, some of the smartest people on the planet are really weird, and they don't manage their social lives properly. And I mean, scientists are famous for this, especially physicists and and mathematicians, perhaps. And so, I mean, you don't want to iron out all the individual differences that make intellectual life worthwhile. So you could say that some of the increased diagnosis is simply a public recognition of a problem, an attempt to medicalize normal individual differences. On the other hand, we have hypothesized and we have evidence for the fact that part of the social aloofness of autism, displayed by many more boys than girls, 80% boys, 20% girls, actually comes from social anxiety. That it's a well-practiced avoidance response. And the little boy, I'm thinking of a specific video, who at his sixth birthday party is forced to go over and greet the guests to his party, but then he goes back over and sits in the corner and reads his book. That it's an avoidance response, that he has high levels of social anxiety. And this can be documented by recording the autonomic nervous system of the little boy, or older children as well, as they face social situations. They really do get quite anxious. So, there's been a hunt for the autism gene. And indeed, I've studied genes that contribute to autism. There's one called catnap2, which is a gene that's expressed in the nervous system. And increased mutation rate in catnap2 is indeed associated with autism in human beings. On the other hand, it's not one of those magic bullet situations in which you have 2 or 3 genes and that's it. We can explain the larger part of autism by mutations in 2 or 3 genes. You want to hear the miserable situation we're in. We have about 800 genes that contribute, and a lot of those are not carried in lines of people, like from father to son to father to son. They're de novo mutations. And there's an age of parent effect. That is, that even though the eggs in the woman are there all the time, the sperm are being dividing, and each time the sperm divides, there's a chance of a mistake. And so as the father gets older and older and older, the chance of having a mutation that will contribute to autism in one of those 800 genes goes up. So the genetic approach to autism has not yielded druggable solutions. That's what everybody was looking for, was a mutation where we could figure out exactly what it is about that gene, what is the chemical which is being produced, and now can we invent a drug which will make up for the chemical imbalance. It's not that situation at all. You can't make up 800 things to treat each patient. And so I'm in favor of behavioral modifications applied very, very early using psychological techniques in order to keep these children within normal human communities as long as possible and at as low cost as possible. Right now in the States, the lifetime cost of taking care of an autistic patient is about $4 million per patient. So it's really expensive, and we have to do better than we're doing now.
Thank you.
Good evening, Professor Pfaff. My name is Chua.
Yeah, where are you? Oh, yes. Yeah.
Meditation affects our works on our brain, boosts our energy, and make us healthier. How meditation affect our brain? Thank you.
Well, the expert on this would be Richard Davidson, who's a professor at the University of Wisconsin. And he has recorded the electrical activity, the EEG, the electroencephalograph, of Buddhist monks, either when they're meditating deeply with different forms of meditation, or when they're not meditating at all. And it turns out that the most striking changes are in the frontal cortex. And so you can be quite certain that when a person is meditating, his frontal cortex electrical activity is being altered. Now that can be thought to be very important both from a cognitive point of view and also from an emotional point of view. Cognitive, it's because, as I said at the very beginning of my talk, when I talked about decision making, it's the organized activity of the frontal cortex that allow proper decision making and the application of good judgment. When a person is getting senile and making bad judgment, it's frontal cortex that's going. And it used to be when people got syphilis, when guys would do things they're not supposed to do and would get syphilis, it was their frontal cortex which was leading to bad judgment. On the emotional side, if there's damage to the left frontal pole, the person altogether sees the happier side of life. If there's damage to the right frontal pole, the person sees the gloomier side of life. So change of electrical activity in the frontal cortex has both cognitive implications and emotional implications. As you were explaining to me before the talk, clearly meditation can do a lot of people a lot of good. And it doesn't cost anything.
I have another question for you related to orexin. Orexin. When there's a mutation in orexin, you were saying that the arousal goes off, the generalized arousal goes off.
No. When there's a mutation in the orexin gene, which is called by some groups the hypocretin gene, orexin fosters arousal. And most people who study orexin for a living would say that it's especially important for organisms that are asleep or not aroused, and they don't have adequate nutrition, and they should get up, they should activate themselves, and they should go look for food. But mutation in the orexin gene will lead to narcolepsy. And that's where the people are happy at the party. I don't know why this is the case, by the way. They're happy at the party, they're laughing, and all of a sudden they're lying unconscious on the ground. Yeah.
Well, what happens to their sexual arousal? Do they go to sleep while having sex?
I don't have the faintest idea. I don't know of any connections between the orexin gene specifically and reproductive biology. It's mainly with the nutrition.
Any more questions?
Yes. Yes.
Hang on. Just get the mic.
You had one slide where you had the neural circuit and you had one where estradiol acts. I see that the effect is also onto the midbrain reticular activating system. So coming back to the clinical application of this slide, I'm also aware that there is some papers that say we can use estrogen in traumatic brain injury. What's the application? What's your opinion on this?
So, two things to say. The first would be that some of the applications of hormones to traumatic brain injury actually have to do with fluid deposition. A lot of the problems after the traumatic brain injury, where it may be in military circumstances or in athletic circumstances, are due to fluid accumulation on the brain, which means that the blood is not getting to the neurons. I mean, you know this, but I'll say it for the group. The blood is not getting to the neurons where it's supposed to be getting, and instead it's causing a fluid pressure and killing other neurons. So it's a double load. It's a failure of neuronal nutrition, and it's a killing of neurons due to fluid pressure. And the steroid hormones tend to be good at reducing that, especially progestins. I'm convinced, the reason I could say that I'm convinced that sooner or later doctors will figure out how to use, and we talked about this before, how to use these NGC neurons to help in cases of disorders of consciousness and to help in the recovery of consciousness, is because viral vectors can be used to deposit into the brain in specific locations and be under the control of specific genetic promoters, so that they'll be operating only in specific groups of nerve cells. And it could be done with extremely small needles into the brain with extremely small volumes deposited in NGC. And I'm predicting that if we were to meet in this theater 50 years from now, that there will be viral deposition into NGC and other parts of the arousal system, which will heighten arousal to make up for the loss due to some kind of traumatic brain injury, especially loss to thalamocortical circuits. But it's not 50 years from now, and so we can't do it yet.
Thank you.
Recently, there's scientific papers linking the central nervous system to the immunity system.
Yeah.
Can you elaborate on that and implications?
Yeah, so it's time to admit that basically I'm afraid of the immune system. In my career, I've had to learn a tremendous amount of neuroanatomy and neurophysiology, and then as the science of genetics came along, I've learned a lot of molecular biology. And of course, we have to know behavioral biology. And then Professor Ishwar and I are expert in the endocrine system. So we already have a complicated system, the brain, and we have a complicated endocrine system, and we know a lot about the relationships between these two parts of the body. And now along comes the immune system, which I'll say several things about. But first I'll make a little joke, which is a true story. There's a great professor of immunology at my school. His name is Jeffrey Ravage. And he's an immunologist. And I was leaving the campus one day, and he was also walking his dog, which was licking my hand all the time, which I didn't like. And I said to Jeffrey, I saw a notice of an immune system talk today, and I actually understood some of the words. And his immediate response was, we're going to have to change the terminology. As though it's like a union shop. It's like a secret guild. Because for all of the specificity that we talk about in neuroanatomy, and I rattle off these long words like the central thalamus or nucleus gigantocellularis, the immune system has even more of them. And I have a hard time keeping track of them. So now to answer your question in a more straightforward fashion. It used to be thought that the brain is a privileged immune compartment. That is, that things that happen with the immune system elsewhere in the body do not happen in the brain. And I talked about the blood-brain barrier and how important that is for safeguarding the brain from all kinds of immune reactions, inflammation, and so forth, which otherwise could screw up the brain. But it turns out there's an oversimplification from at least 2 points of view. The first is that there are immune cells in the brain. One of them we've known for quite some time, and they're called microglia. But recently, a second kind of immune cell called the mast cell, these cells are in the brain. And finally, a professor at my school won the Nobel Prize for discovering dendritic cells, which are immune cells, and dendritic cells are in the brain as well. And they respond to environmental events. So that was an oversimplification to say that the brain is an immune privileged place. Nothing immune happens in the brain. The second reason it's oversimplification is that the blood-brain barrier is not as total as we thought. Some things get across the blood-brain barrier, like B cells. That's one particular kind of immune cell that I've memorized is B cells, that they can get into the brain. They're manufactured in bone marrow, they proliferate in bone marrow, they get across the blood-brain barrier, and they proliferate in brain. That's been shown within the past 2 years. So, we can come at your question from the opposite point of view. How could it have been that we said that the immune system and the brain are totally separate from each other when we know that there are neuroimmune phenomena that you referred to? There's things called sickness behavior. And some of the sickness behavior that's demonstrated by AIDS patients are a proof that there must be neuroimmune phenomena that affect behavior, and there must be mechanisms for it. But the exact mechanisms have not been discovered. So we know now that there are immune cells in the brain. We know that the blood-brain barrier is not totally intact, that some things can get across from it. And we know that there are behavioral phenomena which speak of linkages between the immune system and the brain, but we don't know exactly how they work. Some people would say that the fatigue syndromes are also examples of immune phenomena. So in females, that would be chronic fatigue syndrome and fibromyalgia syndrome. In males, that would be Gulf War syndrome. Some people would say that they have an immune component, but that's not been proven. Now, what part of your question have I not answered? I mean, because you asked a very broad question. Did you have something specific in mind? Yeah. I think, so if I can think of a Venn diagram and have 3 huge circles, the nervous system and the endocrine system, we have pretty good control over. But the third circle, the immune system, which also links the entire body, is not as separate, either from the endocrine system or from the brain as we used to think. But the manner of interaction remains to be discovered.
What's the best way to treat Parkinson?
Remember, I'm not a medical doctor. The best established way that I know of for treating Parkinson's is electrical stimulation of the subthalamic nucleus. If you look at the work of Joe Lemaines at Emory University, they have patients with electrodes in the subthalamic nucleus for 10 years. And the parameters of the stimulation to reduce the involuntary movement remains the same for 10 years. But it's at best, it delays the onset of Parkinson's disease. So across the street at Memorial Sloan Kettering Cancer Center, there's a man named Lorenz Studer, S-T-U-D-E-R. And as you know, it's the death of dopaminergic neurons in a specific dopaminergic system that causes Parkinson's disease. He is making dopaminergic neurons from embryonic stem cells, and he has delayed the onset of Parkinson's disease by implanting these artificially produced dopaminergic neurons in the brain. So right now, the fix is electrical, but in 10 years, I think the fix is going to be embryonic stem cells, and every which way is going to be used. Combinations of therapies are going to be. Levodopa, as you know, has its limitations. So there will be electrical, there will be pharmacologic, and there will be stem cell applications in coming years.
So I'm Alan Lee from School of Pharmacy. So I'm just curious to ask, say, for animals that hibernate, is there anything special regarding the regulation of their arousal status?
Yeah, that's really interesting. I've actually seen squirrels that hibernate where their body temperature's gone way down and they're giving only one breath about every minute. But if you just touch them, they will start to respond within 2 or 3 minutes and start to come out. How does it work? I don't know. Actually, I don't even want to know. I don't even want to know because it takes too long to study. We were talking about development of systems that respond quickly, where I think you have an aging fish, where the fish ages very, very rapidly. And we spend a lot of time in the laboratory. And if you have phenomena which happen relatively quickly, such that you can get an answer, let's say within a week or within a month, you're in relatively good shape. But when you have a phenomenon that lasts all winter, well, that's a little slow for my metabolism. I mean, nothing in New York is as slow as that. Yeah. There's a question up there. In the middle.
Yeah, that can be a PhD project.
Yeah.
Good evening, Professor. Is there a way that we can manipulate the states of general arousal to produce something like a more sustained behavior like motivation?
Mm-hmm. That's a good and practical question. So unfortunately, a lot of the ways of manipulating generalized arousal these days are pharmacologic, and they include the use of drugs which are then misused. It's rewarding to have that rush, apparently, and it's rewarding also to get a relief from rush, let's say by opioids. And the phenomena of drug addiction speak to the attractiveness of manipulating arousal systems. And so I would recommend everything that we see in the motivational programs on television and in the popular literature. One of the ways of manipulating arousal systems is by exercise. I mean, you have to have a higher level of arousal in order to exercise. And for many people, elevating, especially if you do it in a way that you determine rather than somebody telling you to do, both elevating a level of arousal through the beginning of the exercise and then getting a relaxation at the end is itself rewarding. Many people will say that setting very large challenges leads to a level of engagement which is itself rewarding. And so that would be that you have decided by conscious decision to pose yourself a meaningful challenge which will require that you maintain what we would call in slang terms the mojo, the energy, the emotional energy to face that challenge. And so in a sense you've manipulated your own arousal system in order to have a pleasurable and useful experience. But I'm not a clinical psychologist, and nobody comes to me for advice.
Okay, I think we'll like to thank Don for his beautiful lecture today. Thank you very much. It's a great talk. So I'd like to invite Prof. Mahendran to give a token to Don. Please. Thank you very much. Thank you. Thank you, Don. Thank you very much. Thank you, everyone. I think dinner is served.